Here is the last section of protein synthesis, and this is the steps that take place in translation. And I'm going to simplify them a bit so that we don't get bogged down in the details. As long as you understand the basic idea of this, you'll be doing all right. So the different stages of translation have the same names as what we had in transcription. There's an initiation phase, a elongation phase, and a termination phase. But the thing to remember with both transcription and translation is that they all require energy in the form of ATP, or as we'll see in a minute, ATP's crazy cousin GTP, which is guanosine triphosphate, but it still is a type of energy. So in the initiation stage, there are three things that have to happen. First of all, the small subunit of the ribosome has to line up with the mRNA, and not just anywhere on the mRNA. Remember, we always want to start with the start codon, which is AUG. And the way that this happens, it's not that the ribosome can see or recognizes like, hey, you're the start codon. It's that the AUG, remember, anytime you're talking about molecular interactions, you're talking about shape, size, and charge, or polarity. So there's a shape and size connection between the small subunit of the ribosome and the AUG. That's how the small subunit knows to line up with the AUG. So after the mRNA and the small subunit join together, the next step is that you bring in the tRNA, remember the transfer RNA. The transfer RNA transfers the amino acids to the growing polypeptide chain. And the anticodon, because the start codon is AUG, the anticodon on the tRNA has to be UAC, and that corresponds with methionine. If you look back in the chart, AUG, the start codon always codes for methionine. So that's the second part of initiation. The third part of initiation is that the large subunit comes in and completes the ribosome. So here is a picture of what it looks like. The tRNA doesn't really look like a T. We're going to use a couple of different types of pictures, but I like this one best because it showed the how the initiation takes place. So step one is the small subunit lines up with the messenger RNA, gets it in the right spot. Second part is that the tRNA with methionine attached to it, and here's what I like about it, it shows the start codon and then the anticodon on the tRNA. And then, in order for this to be completed, you need energy. Here's the GTP that I just mentioned. And now you have the completed ribosome that is all set up and ready to go. Don't worry about these, I don't know, what do they look like? Hot dog? End of a hot dog sections. We're not going to add that into our discussion. So the next step in translation is elongation. This is where the protein is made, where the amino acids are linked to each other. Let me go through it with the words first, and then we'll take a look at the picture. So if you noticed, let's go back a step. If you noticed, this spot in the ribosome is empty. Well, here's 1, 2, 3 bases that line up to that A spot that is empty. So that's going to be a spot where the next tRNA can come in that has the correct anticodon, which is going to bring another amino acid to sit right in this spot next to the methionine, and that's going to line it up so that the two amino acids can attach to each other. So that next tRNA with the correct anticodon is going to go into the spot right next to the methionine, and it's going to have the right amino acid on it. And the methionine, whatever that next amino acid are going to be bonded together with a covalent bond. And remember, anytime you're talking about an organic molecule, and you are creating a covalent bond, the name of that process is dehydration synthesis. So this is happening through dehydration synthesis, which requires energy. And here's our picture. It's a different set of pictures than what I was using in the first one, but you still see here's the messenger RNA. There are bases in front of the AUG, but we're starting here with the AUG because that's the start codon. This tRNA happens to look like an Erlenmeyer flask, but it's got methionine attached to it. The next codon is UCG. The anticodon that goes with that is AGC, and you could look at the chart to see what amino acid that's going to be. So that amino acid, that tRNA with the amino acid comes into this spot. And then the methionine is hooked to the next amino acid in the chain through dehydration synthesis. And now you have a tRNA that no longer has an amino acid attached to it. And the next step in the process is that this ribosome is going to slide down one spot so that the process can continue. So in this case, now the tRNA that no longer has an amino acid attached to it gets kicked out. The ribosome has moved down one spot. If you want to think of that, if you see this E, if you want to think of this as the ejection spot, that's where the tRNA gets kicked out. But now there's an open space for the next tRNA to come in. And that's what this is showing. Go ahead and pause the video if you need to write this down. All right. So this process continues. The next tRNA with the correct anticodon and the correct amino acid enters the ribosome, and this continues until a stop codon is reached. That's the end of the elongation process. Last step is termination. In the termination step, instead of a tRNA with an amino acid, when you get to one of those stop codons, it's not a tRNA, it's a release factor. It's another type of protein that basically creates that roadblock like we talked about in transcription that causes the whole thing, the whole ribosome, all the pieces and parts to break apart. The last amino acid in the chain detaches from its tRNA so that now you've got a protein that is flying free. Ooh, that's some bad animation. The amino acid chain is released and the ribosomal subunits separate from each other. So here you've got these purple shapes are the amino acids, the protein. This is the last tRNA. Here's where your stop codon is going to be, either UAG, UAA, or UGA. And remember, that'll always tell you that in the chart. And you will always have the chart. You don't have to memorize the chart. So then the release factor comes in, and that causes everything to just break apart. And now you have your protein that can go and do its little protein job, live its little protein life. The large subunit and the small subunit of the ribosome break apart. The release factor goes on its merry way, and the messenger RNA is released. And depending on what type of messenger RNA this is, that messenger RNA could be translated again. It could be broken down into parts and recycled, or it could be used just a couple more times, making the protein for a short period of time. All right, so that is translation. These 3 video links, yes, I want you to watch all 3 of them. It's going to be a lot of repeat information because transcription and translation doesn't change. But each one focuses on a slightly different part of it, and it's important that you watch all 3. You can do that on your own time. I'm going to continue right now. I've got a couple more slides to go. So let's talk about a process called wobble. I don't know if you noticed in the chart that we had, but a lot of times if you change the third base in a codon, there's going to be no change in the amino acid. And you can go back and look at that chart again. But for example, it doesn't matter whether you have GCU, GCC, GCA, and GCG, they all code for the amino acid alanine. So that third base in the codon doesn't really matter. There are only two amino acids that have to be very specific. One is AUG, that's methionine, that's the start codon. And the other codon that is very specific is for tryptophan. And I don't remember off the top of my head what the codon is for that, but everything else has at least two codons that code for the same amino acid. This idea of the third base not really mattering a whole lot is called wobble. So that third base can wobble, it can be different, and you still get the same amino acid. All right, now let's talk for a second about mutations. We've already talked in class about how mutations are changes in the genetic information of a cell. It's changes to the DNA. A couple of terms. Mutagen is any substance that can cause a mutation. That's a little bit different. You've probably heard the term carcinogen before. A carcinogen is any substance that causes cancer. Mutagens can cause mutations, but they don't necessarily lead to cancer. Some substances that are mutagens could be ultraviolet radiation, it could be environmental toxins, could be just any type of substance that can cause a change in the DNA. But the results, this is going to sound really familiar because we talked about mutations in terms of natural selection. Most of the time, mutations are neutral or silent, and that has no impact on the protein that's produced. Sometimes the mutation causes a decrease in the functioning or efficiency of the protein. So it doesn't cause the cell to die, it doesn't cause cancer, it just means that the protein, whatever it is, maybe it's something like a digestive enzyme. It's just not as good at doing its job as what it should be. But it is possible that some mutagens can result in disease or death. And that's a real quick fly through mutations, but it gives you the basic idea. So there you go. That is protein synthesis in a nutshell. Let me know if you have any questions.